Good morning, everybody. Good morning. Thank you for coming. Nice meeting you. Say that it's just okay. Okay, so it was on me. Maybe. Got it. Okay. So, I'd like to introduce Dr. Chambers. Dr. Chambers earned her Bachelor's of Science in Natural Resources and Conservation Management from the University of Botany. She earned her PhD from Purdue University in Botany and Plant Pathology and did her postdoctoral work at the University of Florida. She's currently a research botanist at Ronald L Botanical Gardens, located in Sarasota, Florida. While in Sarasota, Dr. Chambers is also the teacher at the Ringling College of Art and Design and the Blue College of Florida. Dr. Chambers specializes in fern biology, especially for endemictites, although she's also worked on the conservation ecology of cacti, cycads, and bromeliads. She has authored or co-authored 19 research publications and is a recipient of an early career grant from the National Geographic Society. In her research, she utilizes various molecular and physiological techniques to understand species distribution patterns, community assembly, population interaction, and how these factors may shift in response to global climate change. Please join me in welcoming Dr. Chambers. Everybody, thank you, Dr. Cosmic, for the lovely introduction. It was a pleasure to be back in my home state of Kentucky. I'm native of Owensboro. So thank you all for coming to my talk. And I really want to address utilizing, thank you, utilizing field biology and collections in my research. I want to give you a brief overview, this is the outline of my talk today, about how I use field biology and collections in my research program. I'll specifically talk about the study systems that I focus on. So as mentioned, ferns, and then I'll give you a highlight of some of the conservation work I've been doing with the rare cactus. And then give you some research examples of field-based studies, laboratory manipulation, and how I utilize and contribute to collections. And then finally, I will talk about the research that I would like to do here at EQU. All right, so first things first, my research program. My big question is really understanding what drives species distribution patterns and niche differentiation. So we can think about this as a broad scale question focusing on species distribution patterns, right? So narrowing that down, I focus on populations. So how do populations interact with our species geographic ranges? How do they differentiate from one another? So we can again think about this as going from the big geographic range down into individual populations that comprise the species geographic range. And then finally, how can we use this information to inform conservation efforts? So this is really kind of a genetic-based or an individual-based question, okay? So again, going from broad species distribution patterns to populations, and then all the way down into individuals or genetics. So I integrate three kinds of arms to my research program, so ecology, physiology, and conservation. Specifically with field and manipulative biology, these interact with ecology and physiology by using species or populations and studying how they interact with their environment. And then this provides practical information for applied conservation projects. In terms of collections, I document and inform location efforts or find populations, using ecological information. So basically collection information can help me find populations to do this ecological work. Collections are also important for conservation, so we can use that to preserve DNA, spores or seeds, and also living material by depositing them as ex situ accessions. And then also data obtained from these living collections can be used for physiological analyses. So basically having access to some of the populations, we can do physiological experiments on those. Okay, so moving on to my study systems. If you're not familiar, this is the general fern reproductive life cycle. So the first question you might say is, well, live ferns, right? So things don't really eat them, humans don't eat them. Why do you care about ferns, Sally? All right, so they are the only lineage of plants where there are two life stages that exist freely and independently of one another. So here you can see this. We have the metatophyte life stage. This is haploid. This is the life stage that produces a sperm in the egg, okay? Once the sperm and the egg unite, then we have the sporified life stage. So then we have this diploid condition. In ferns, this is probably what you're most familiar with. But again, in ferns, these actually live independently of one another. The gametophyte is completely photosynthetic and capable of living entirely on its own. All land plants have this reproductive cycle. We call this the alternation of generations. Ferns are the only lineage of land plants where these actually exist independent of one another. So in terms of understanding land plant evolution, ferns are pretty critical in that, right? So they evolved after bryophytes, before angiosperms and gymnosperms. So by studying this alternation of generation in ferns, we can really look at detailed questions in land plant evolution. So this is the typical textbook depiction of a fern life cycle, and specifically of the fern gametophyte. Okay, it's this cute little heart-shaped organism, but in reality, fern gametophytes are much more complex morphologically. So here is our little coordinate fern here, little heart-shaped gametophyte. You can see there's all kinds of different morphological conditions in which these organisms can exist. So they're a lot more diverse, kind of to a degree. You can see that there's still not a whole lot of morphological variation. They do vary, they're not just heart-shaped, but in terms of identifying them, you may not have a whole lot of characteristics to go off of. So and you might ask yourself, okay, Sally, you told us why you like ferns, but why do you like gametophytes? And actually, even amongst the fern community, they were once considered to be like the handicap generation of the fern life cycle. But a lot of us in the fern community now realize that's not true, and we understand that you actually could not feasibly have sporophytes if you do not have gametophytes. Gametophytes inherently, through their fertilization, through the fertilization that occurs in the gametophyte generation, that's what produces sporophytes. So you cannot have them if you don't have a gametophyte previously. So also, by studying the gametophyte generation, there's a lot of comparative biology that I can do. So I can compare, stress tolerances in the gametophyte generation to stress tolerances in the respective sporophyte generation. And gametophytes are really easy to grow through spore and even maintain in culture in a laboratory condition, and they're great for manipulative experiments. I'll get into that in a little bit. All right, so ferns also reproduce in a variety of ways. So again, going back to that typical textbook depiction, what you're observing is called gametophyte stumping, right? So you have one gametophyte, you have a spore in it ignite, and then that produces a subsequent sporophyte. In reality, you can also have sporophytic sulfing, where the same sporophyte produces different gametophytes, and then those gametophytes cross, okay? And then have a fertilization event. And then we can have outcrossing, where we have individuals from different sporophyte parents that are then interbreeding and producing a sporophyte offspring. And there's even asexuality that occurs in the gametophyte generation. And so I want to introduce you to Botania appellatiana, commonly known as the Appalachian shoestring fern. This is a picture of this little cutie, and this is a close-up of some of the microscopic conditions, and these are the asexual properties that Botania appellatiana produces. And we'll talk a little bit more about that in a minute. Okay, so going back to this concept of the difference between the sporophyte and the gametophyte life stage, there's also difference in stress tolerances and environmental gradients that these two life stages can experience. So we can have conditions that are favorable for a sporophyte, conditions that are more favorable for a gametophyte. Obviously, there's a whole lot of overlap between these two generations inherently. And we'll get into some of that again, some of my research in a little bit. And I also want to introduce you to the cactus, the rare cactus that I've been studying in Florida. So this is Herbarium aboriginum. It is a federally cactus. This is a picture of it. It's on here. It's actually pretty tall. It can get over 6 feet tall. And this is a picture of the fruit in my hand, actually. And then this is a picture of the geographic range, right? So it's very restricted. It's found in these coastal communities and very few populations live in existence. And I'll talk more about some of my research about that as well. So moving on to research examples, field-based studies first. Okay, so going back to Botania appellatiana, this is a species that I actually studied for my graduate work, and I really wanted to examine plant responses to climate change. So the first thing that I did was look at local adaptation. And you might ask yourself, well, what is local adaptation? So that means that populations, individuals from one population, are going to be best in their home population, their home environment, If you move them to another population where the same species occurs, you're not going to do as well. Right? So they really prefer big companies, right? So the way that we test this is by taking individuals from different populations and then transplanting them, right? So this is just the arrows depicts where I have moved individuals from Bacteria Appalachiana. So this green represents the geographic range. The people star here is where you're located in Richmond. So you're basically right in the heart of Bacteria Appalachiana country. And again, these black dots represent all of my sample populations, and I just did this reciprocal transplant by taking individuals and moving them amongst these different populations and examining their performance. So this is kind of a picture. It's not too great. I'll tell you a little bit why, but this is a picture kind of my experimental setup, and you can see there's individuals represented from each of these populations in that transplant block. So basically, what did I find? What were the results from this experiment? Well, I didn't find patterns of local adaptation. Actually, what I found was a completely different pattern. So just to take a subset of my data, I took individuals from these different populations and I moved them into the population in North Carolina and I examined their performance. So how many survived after a year of being out in the field? And what I found was that actually individuals from New York performed better in this North Carolina population, and that was apparent in all of the other populations as well. So if we expected to see local adaptation, what we would find is the black line, which represents the individuals from North Carolina, would be performing better up here, right? So these lines would be flip-flopped. So instead of finding this pattern of local adaptation, I ended up finding a pattern of counter-gradient selection instead. So what that means is that individuals from New York are going to do better in any of these other subsequent populations. And that's important because that helps us understand which populations may be good for conservation purposes in the future. Okay. Following that same kind of line, right, I wanted to understand if the species could move north in the future, right? So that's one of the ways that we think species will respond to climate change is by shifting their geographic ranges. So I identified a population that was just beyond the northern range boundary. Right here, it's depicted by this little gray dot. And again, I did a reciprocal transplant. So I took individuals from each of these populations and I moved them to this location. I also monitored the microclimatic conditions of this population to make sure that they were suitable, and they completely were. And what I ended up finding is that there is a social limitation that drives its northern range boundaries. So basically, this species is not going to be able to move on its own. If we want to do something to conserve the species, we have to physically move them ourselves. And part of the reason why this occurs probably has to do with the asexual reproductive organs of Bacteria Appalachiana. So these genite, you see here, that's what they're called. These kind of like little sticks of 5 or 6 cells. Those are the asexual reproductive units, and they're definitely larger than the spore, right? A fern spore can get up in the atmosphere and travel literally thousands of miles. These are not going to be able to do the same thing. So the fact that this is the only reproductive mechanism in Bacteria Appalachiana is probably the reason why this dispersal limitation is occurring. And again, so what I found out was that individuals are capable of surviving in this northern range boundary, beyond the northern range boundary. So there is suitable habitat, they're just not able to get there, right? They're not able to access it and colonize it. And it's important to documenting changes and how plants are going to respond to climate change is really understanding the climatic conditions that they're coming in now. And this also gets back to this concept of the fact that the sporophyte and the neophyte can have these differences in their environmental tolerances. So being able to monitor these climatic conditions on a time scale where we may find a sporophyte and we may find a neophyte can help us do some of this comparative biology. So this picture of, we're now moving from Bacteria Appalachiana to a different study system. This is Calisandrus Botany. This is a filmy fern that is found on the island of Botany in Hawaii. And the interesting thing about this plant is that it's got this really cool elevational distribution. So at the bottom of the mountain, you find the neophytes only. At the higher elevations, you end up finding the sporophytes. So I worked with a colleague to go out and deploy these very fancy climate sensors and determine if we are seeing differences in climatic tolerances of the sporophyte and the neophyte generation. So what we did find is that there is life stage variation, and this is actually kind of a beautiful like stair-step pattern, right? Where we can see that the neophytes can tolerate warmer temperatures, right? So at the base of the mountain, it's going to be warmer, and then at the summit are going to be able to tolerate, some of these cooler conditions only because they're at higher elevations. So the next step, if we could have done this, would be to take some of these neophytes, take some of these sporophytes, and do this manipulative work to expose them to different climates and really see how they respond to kind of nail some of that down. So in addition to doing field-based science to examine responses to climate change, I also dabble in molecular ecology. And going back to this concept that, the neophytes do have morphological variation, but it's only to a degree. There's plenty of examples where you find a neophyte in the field, and there is no way that you can identify its species. If you're lucky, you could place it in the genus, you could place it in the family, but the morphological characteristics to distinguish them just aren't there. So we have to use molecular tools to get a species identification on these taxa. So a colleague of mine and we were told about some really unique gametophytes that were found in Georgia. And so we went out there and we made these observations, hand lines to give you some scale. This is Botania graminifolia. So this is a relative of Botania appellatiana found here in the Appalachian Mountains. But graminifolia is actually occurs in Florida and throughout the tropics. And then this is Gametoglossus petersii. So the only reason we were able to put a name on these species was by actually using molecular tools and doing some barcoding. So this is important because this is actually a new record for Georgia. The species have not been reported there before. And it was also exciting because, given the fact that this was located in Georgia, kind of in the middle of where we find Botania lineata and where we find Botania appellatiana, we didn't know if this would be Botania graminifolia, we didn't know if this would be Botania graminifolia, Botania lineata, or Botania appellatiana. We really needed to go and collect this individual and be able to put an identification on it, because if it was Botania appellatiana, that would be the first occurrence of finding a sporophyte of this species, or mature sporophyte of this species in the field. And so I also want to highlight a unique thing about these taxa, right? The Gametoglossus petersii actually occurs throughout the tropics. As do a number of these really unique neophytes found in these rock shelters. So you can see that these are the distinct populations up here that occur in populations in these rock shelters. This was the new record that we found. Lineata occurs down here and throughout the tropics, and then graminifolia is actually down here as well. Graminifolia is actually the sister taxa to Botania appellatiana. So a lot of really interesting patterns and things going on here with neophytes and sporophytes and different taxa, and utilizing molecular tools to even be able to get a name on them. So moving on to my conservation research, understanding the current status of the species is really important, right? So if the species is determined to be rare, we really need to understand how many individuals are left in the field, how well those individuals are performing, to allow us to kind of then create a conservation management plan, if you will. So based on Herbarium aboriginum, we have this. It is very rare. It's found in 3 counties in Florida. It's actually extirpated from 1 county, and that was the county that was used to find the species, to name the species. So no one had really gone out and done a thorough analysis of how these populations were performing in about 10 or 15 years. And there have been a number of hurricane events that have happened since then. And given the fact that this species is such a coastal plant, we really needed to get back out and see, have these storm surge events, pull plants out of their habitat, has urbanization impacted them further, things like that. So my colleague, Dr. Sean McCourt, and I, we went out and visited all of the extant populations. And unfortunately, what we found is that there has been a significant decline in the populations since their last thorough analysis. So there's been, more than 50% of the individuals that have become extirpated in the past 15 years. And I want to just highlight here, these were the previous surveys, and these were all of the locations that we visited. So you can see, for instance, the Minnesota Beach, they significantly declined from about 125, and now we've counted 31 individuals. Some of these locations, actually had, some individuals, so one of the water club, there were 5 individuals in these surveys, and then it's run down to one individual. In this particular instance, this decline is specifically due to the fact that a condominium has been developed on the site. Prior to these analyses, a New College of Florida student had gone out and done some surveys, and she counted over 100 individuals at that site. So this condo went in and they said, oh, we have this rare cactus, that's great, we'll subset, 4 acres and conserve it. And then they divided those 4 acres into 2 plots of 2 acres, and then they just didn't manage it. So we went back to the individual, and unfortunately, this one individual that was remaining has since died. We tried to do an ex-situ rescue, and we could not maintain the individual. It was too far gone. We were able to collect it. So unfortunately, the species is no longer located at the Longboat Key Water Club. But we did find a few new populations, but again, there are just few numbers of those individuals in those locations. So another important thing to look at when you're doing these demographic analyses is to look at the reproductive potential. So we find individuals, that's great, maybe we find a lot, maybe we find a few. Are they able to produce fruit? So we also wanted to look at the phenology. So basically examining the number of flower buds and the number of fruits that we see being produced. And we found that only 21 plants that we examined were reproductive. So that's not very many. And we also didn't really observe a lot of seedling recruitment at these sites either. So that tells us that there's not a lot of natural recruitment that's happening in these populations. We're losing them at a greater rate than they're able to reproduce and repopulate. So another reason that they're probably super rare. So following this, the end goal of this is to really conserve the species and to bolster the numbers that we see in the field. So we have to do some testing to figure out, what are the best conditions in which to transplant these individuals. So there's a few factors that we wanted to test. One of them was the age class. So basically, if we put seeds out in the field, are they going to do well? If we put seedlings out that are about a year old, do they do well? Do we have to grow plants to 5 years for them to be big enough to be transplanted and to stay out on their own? Because we don't want to be going back and augmenting these populations repeatedly. We want to set them up to be successful, on their own in the long term. So we tested age classes. We also tested different light conditions because this cactus is what I refer to as a Goldilocks. So it doesn't like a whole lot of shade, doesn't like a whole lot of light, basically likes to be right in the middle. So we wanted to test that thoroughly. And then we also wanted to look at the impacts of the woodbury, right? So that's why we have these big cages here. You can see the cacti transplanted here in some of these little pots. But this is a study that's in progress. I can tell you some preliminary results. Seeds don't do well. We had one seed germinate, and then the seedling died subsequently. And what we're seeing is actually kind of a flip flop. So if we looked at data, that were 6 months out from transplanting, then we would say, okay, individuals that are 2 years old are going to do the best. And move that fast forward to a year and a half, we see that actually individuals that are 5 years old are doing the best. So this is why it's important to continue long-term studies and things such as that. So again, this is in progress. It's just a pilot study. It's to help us gain information so that when we go and actually do an augmentation or a reintroduction, we know the best conditions in which to do that to set these individuals up for success. And I also want to mention that this has been funded by a number of organizations, so Fish and Wildlife Commission, and Florida Native Plant Society, Yankee County, and then the Cactus and Succulent Society of America. Okay, moving on to laboratory manipulation. So doing things in the field is fantastic, but sometimes you do these things in the field and they might pose additional questions, right? So for instance, I found this pattern of pure ingredient selection. What is driving that? So I went with a colleague and learned how to do some stress manipulation experiments, essentially. So I took individuals from all of these different populations again, and what I did was I exposed them to different relative humidity conditions and their ability to recover from that. So these are all these little neophytes here. You can see Alabama and all of these different conditions, right? So they were basically elevated above saturated salt solutions. So this is what I'm going to write about on the bottom here. And the salt solutions allow us to create different relative humidity conditions. So I could create a 12% relative humidity condition in this tiny little plastic box. Okay, so we can test 12%, 75%, 97% relative humidity. And then obviously compare that to control. And we use a chlorophyll fluorometer to do this. So this is the chlorophyll fluorometer. It essentially shines a bright pulse of light, and it gives us a metric to understand photosystem health. So you take a baseline reading, and these baby ferns, these neophytes, are really happy, stress them out, and then look at their ability to recover to that baseline condition. So this is what I ended up finding. We can see that we're looking at the proportional fluorescence here. So what this means is that this is the amount of that original photosynthetic capacity that individuals were able to, get back to after being stressed out. So across all the treatments that I tested, we can see that New York actually does the best. So these individuals are able to get back to their original photosynthetic capacity at the greatest rate compared to the other populations. So this actually most closely reflects the pattern of counter gradient variation that I found by doing my local adaptation experiment. So it's also important to look at temperature tolerance, right? Especially when we're thinking about how plants are going to respond to climate change. We know that there's going to be variation of precipitation patterns. So that's going to be, affecting desiccation tolerance. And then also temperature tolerance, right? So if temperatures are elevating in the future, how will our plants or different populations going to respond to those elevated temperatures? So again, I took those individuals that I had collected in the field, and I stressed them out. So I built these little boxes in this one growth chamber. And looking back at this, it kind of looks like it could be a fire hazard, but the facilities people assured me it was okay. And so I built these little chambers, and I basically had these heat mats at the bottom of the chambers, and I elevated the plants, to different levels, so that they were experiencing 10 different temperatures within one growth chamber. And what I ended up finding is that there's actually a striking similarity of all of these populations in their ability to tolerate temperature conditions. So these are the 10 temperatures that I tested, all the way from basically 5 degrees Celsius to 3 degrees Celsius, so a very wide range. And you can see a significant decline in performance, a little bit before 15 degrees Celsius. And then again, these are the populations, and overwhelmingly they essentially respond the same to these thermal conditions. So basically, we can see that as a species, Botania acretiana has a similar temperature threshold, but they vary in their relative humidity thresholds and their ability to tolerate relative humidity conditions. So thermal tolerance is relatively similar across all of these populations. Okay, moving forward with some laboratory manipulation, we're going back to conservation. So going back to some research on that cactus. So again, in the field, we looked at these demographic analyses, right? We see a decline in the number of individuals that are remaining, and again, we saw very little evidence of flowering and fruiting, and very little evidence of natural seedling occurring in the field. So why are we seeing this? Are these seeds able to germinate well on their own? Is there something that they need to experience, like maybe they need to be eaten by a gopher tortoise, and then go through the digestive tract to get some scarification before they germinate? So I had a wonderful student from the New College of Florida that came over and worked with me for a month, and she did a germination analysis. So these are mostly Aboriginal seeds. You can see this is one right here, and these are them germinating. And what we can see, we tested a few different things. So first off, we did a control where the seeds just basically experienced a water treatment. We tested gibberellic acid, which is a growth hormone. We tested gibberellic acid in combination with sulfuric acid. Sulfuric acid represents the scarification of the digestive tract, and then sulfuric acid alone. And so what we can see is that sulfuric acid significantly increases the germination rates of Herbarium aboriginum. And then I just have these in pink boxes because we also tested a few other taxa, but I just want to highlight the Herbarium aboriginum results here. So again, sulfuric acid greatly increases germination success, suggesting to us that these species do need to be digested by something to get a great further germination rate. And I do want to mention that these are data that are currently being written up for publication, and Wes will be a co-author on that publication. So moving on to how I utilize and contribute to collections. I use collections in various ways, and I contribute to them by doing documented field work. So I'll just give you some examples of that. In terms of utilizing collections with my genetic work, then it basically comes down to doing phylogenetic analyses. So in my postdoc, I worked with Dr. Emily Spezza to examine the biogeographic patterns of Dryopteris. So this is a large group of ferns worldwide. There's a number of types that exist in Africa, and we really weren't sure how these species came to arise in Africa. So this is just a really pretty population map of where these different Dryopteris exist. So basically, we used herbarium specimens to constructively sample tissue so that we could get the genetic material to do the phylogenetic analyses necessary. So without those collections, without those herbarium stored collections, we would not be able to do this, right? So what we found by first building phylogeny and then doing a biogeographic analysis, we determined that most of the African Dryopteris arrived in Africa and that way in Asia about 10 million years ago. So they were first in Asia, then they came over to Africa, and then they diversified essentially. So by contributing to collections, I do this in a variety of capacities, but this is kind of one area that I want to highlight. I visited the island of Dominica, basically before the world shut down for COVID. And basically, I went to Dominica. This is my geothermal project. If you're not familiar with Dominica, it's a very tiny island in the Western Antilles. It's not the Dominican Republic, it's a completely separate place. It's called Nature Island because there's 70% of the intact vegetation remaining. So it's one of the most vegetated islands in the Caribbean. And it's also really interesting in its location because this is one of the first to experience trade winds that come up in Africa. So that could be a really interesting community assembly pattern that we see. It's also very steep in terrain. So going up in elevation, you experience drastic changes in community composition really quickly. So my question was, does phylogenetic clustering change with respect to community assembly along these elevational gradients? So essentially, are these communities driven by either ecological preferences or are they driven by their evolutionary history? So this is in progress. This is a publication I'm currently working on. But to do this, to even build the phylogeny, again, I had to get the tissue. So I visited Dominica, I made a number of collections. This is a very weirdly isolated island, so there's not a lot of collections that even exist in our area across the country. So I collected these individuals. I left a specimen there in Dominica, and then I brought the rest with me back to Slowly. So this is what the field looks like here. This is an open forest. It's one of the highest elevations on the island, and this is one of my favorite collections. This is Herbarium or Quaesitis, and you can see that it's yellow on the underside, right? So this is a substance called farina. It's pretty fun because you could, put it on your friend's arm and give it a little smack and really like a temporary tattoo. So it's a fun fern to play around with, and it was really interesting as well because the species is not represented on GenBit. So anything that I generate genetically from this specimen will be the only genetic information available for this species. So in terms of population genetics and contributing to collections, conservation work that I've done with Lucia is really important because we are making the DNA, so we're doing long-term storage of DNA that I'm extracting. If you see, this is the top of one of the cacti individuals, and I'm collecting spines. So I don't have to hurt the plant at all. I can cut into the tissue. I can collect these spines and extract the DNA from the spines that way. So we're collecting this DNA and making it, and we're also long-term storing seeds for conservation purposes, and then also doing ex situ collection. So trying to germinate individuals from seeds, or even maybe sometimes acquiring individuals from the field. So basically, this just points to the fact that living and preserved collections are invaluable. They're really important. And we couldn't do this conservation work without having these living collections. And then, I couldn't do a lot of my genetic work without having access to preserved collections as well. So I also use collections in computational modeling. So I can build species distribution models. And basically by accessing all of the digitized information from Herbarium worldwide, I can then basically map where all of these individuals occur, right? So all of these dots represent occurrence records that come from Herbarium specimens. The Herbarium specimens are really important because you know that they're legit, right? If I take a human observation, it could be that, someone with not a lot of experience put an identification on that. By taking Herbarium records, we can be pretty confident that that's actually the correct individual that we're looking at. So using these occurrence records, we can then build these species distribution models. So we can then basically understand this is T. This is a Stratford found in Florida. This is its distribution based on the species distribution model I built, and it does reflect where we find that species in the state. And then importantly, we can also project this into the future. So we can say, okay, this is where we're finding it now, based on the climatic conditions in the future. How do we think these populations will respond due to these climatic conditions? So we can see that actually, in terms of climatic stability, there's not a lot of climatically suitable locations remaining in Florida by the year 2070. So I also do florescent inventories in my current position. So these are really important because they help us document biodiversity. They help us understand, what's living where, right? So this is kind of the basis underlying emphasis of all of the research that I do, essentially. So a lot of the inventory work that I've done has been in my current position and it's been in tropical areas. So this is just some pictures of the process if you're not familiar. When we're out in the field, we might find an interesting species that we want to collect for that area. Again, we're trying to document the biodiversity of a region. This is a really cute little anemia, and we give it a collection number. We take a number of photographs of it to document it. We get it books in the field, and then we collect it. So we'll basically refer to take a portion of the rhizome, and then any notable features of that plant, we're going to make sure we get in your collection. For this anemia, they're dimorphic fronds. We want to make sure we get the fertile frond as well as the sterile frond. So you come back and you're all excited because you find all your plants. You've had a really fun day in the field, and then you eat dinner, and then you have to do more work, right? So then we are fixing plants. Again, these are going to be turned into preserved collections that are going to be stored in an herbarium. And the important thing about this is that it does help us document biodiversity patterns. And then we can also turn this information into a handy-dandy field guide. So we can make this information freely available to the greater scientific community. Based on the work that I have done with the bees, with my colleagues, and then it's my boss, we have been able to document the epiphytic and metaphytic fern, genera, or Belize. So I turn this into a nice little field guide. If someone goes to Belize and they want to be able to identify a fern, they can download my guide. There's a lot of pretty pictures, there's some great text information, and it's really available to anybody that wants to access that. So it's a really great resources, especially for the broader scientific community outside of the United States. Okay. So moving on to research that I would love to do here at Eastern. Okay, so going back to the neophytes, right? They're tiny, they're cryptic, we can't really identify them. We identify them based on this barcoding process, right? So here we have the sporophytes of a given region. We can sequence them for a specific gene or a couple of genes. We have this unknown gametophyte, and we can basically take this unknown gametophyte and then compare the genetic content to the sporophytes of that area and say, do you match or do you not, right? So this is important for us to understand biodiversity patterns, where these gametophytes are growing, and this is covered in a review paper that a colleague of mine and I just wrote. This is a process called DNA barcoding, if you're not familiar with it. So moving forward, there's the capacity to actually do this in the field, right? So previously, you go out and you collect your gametophyte, you have no idea what it is, you bring it back to the lab. and you do your genetic analyses, and you wait until you wait for something, and then you have your identification. We can now take this portable sequencer called MinION out into the field and get these analyses or this data in real time. So that's very powerful because it will allow us to address a number of ecological and physiological questions that we currently just cannot do because we don't know who anybody is. So we can't study these organisms in the field. And again, understanding gametophytes is really important because you can't have sporophytes if you don't have a gametophyte. I would also like to continue doing conservation projects, so specifically looking at demography and phenology. So this is actually one of my favorite house, plants. So this is Botany and the shooting star. According to the Kentucky State Nature Preserves, there's about 176 endangered plants and 101 endangered or 101 threatened plants in the state. So there's a lot of taxa to work with to do this conservation type work. And again, I think this kind of research provides a lot of questions. There's a lot of opportunities for student research, a lot of hands-on and important applied research as well. And these are some organizations that I think would be great to partner with, where go present data and results for us. So Kentucky Native Plant Society and the Kentucky Nature Preserves. Also going on to this fact of modeling species distribution patterns, not only are they important for looking at the potential for species to respond to climate change in the future, but also the potential to locate new populations. So this is a paper that came out just a few years ago, where this individual modeled a number of plants, and they actually were able to find new populations. So the models worked really well, actually, for this rare species, this rare species. And they were able to locate new populations. So they were able to find new records. So that's really important, right? And another aspect of this is actually integrating this population genetic analyses. So this is actually a paper on crayfish, but we could take the same kind of approach in a number of different rare taxa, right? So you build a species distribution model and then you couple it with genetic variation. So then that helps you understand, okay, we see a lot of genetic variation in this population. Oh, this is going to be suitable habitat. We want to make sure that we preserve this into the future, right? We can also see a lot of genetic variation in these regions, but according to the model, that's not going to be suitable habitat. So we might want to make sure that we have accessions from these individuals, and if you do any assisted migration, we can move them into these new regions, or at least make sure that we're preserving that genetic data. Okay, laboratory manipulations. I would like to continue on this process of desiccation tolerance. Again, because gametophytes are really easy to grow and manipulate. What you're seeing here is actually like a baby gametophyte, and then these are emerging very young sporophytes. And this is great for doing work with master's or undergraduate students. So this is a student that I currently have with me at the College of Florida, and she is actually leading some of this project, where she's growing plants for me, and then here shortly, in the next couple of weeks, I will be teaching her how to do these manipulative studies. And then eventually what we're going to do is get the tissue from these stress responses. So I will generate the physiology data, and then we will then ship those tissues off to my colleague, Wei Li. He's at the Bruce Thompson Institute in New York. And then he will look at gene expression. So people do an RNA analysis. That will help us understand which genes are driving desiccation tolerance, which has a wide range of implication for a number of land plants, as you might imagine. So moving on to conservation projects, again, using these models and just hearing information and working with partners in the state, I feel it's important that we could go out and document new populations. We can also continue with floristic inventories, and I know that this is something that master students have done repeatedly here in the past here at PKU. And then also, I think I personally love these natural communities. Obviously, that's where material is found. But there's a number of really interesting taxa that exist in these fractured communities, and I think there's a lot of opportunity for both conservation projects in these rock shelters, but also building phylogeny and looking at these really interesting patterns of community assembly in these rock shelters, even compared to other regions. So with that, I would like to thank a number of people. I have a whole lot of people to thank. So anybody featured in this has been important and integral in all of the research that I've done. My PhD advisor, my postdoc lab, my great colleagues who highly present at the University of Florida, fantastic colleagues, the students at the College of Florida, and then my amazing colleagues at the Marie's Botanical Gardens. And with that, I would like to thank you, and I'd love to take questions. Yes.
Dr. Chambers, could you maybe help us understand what kinds of teaching experiences you had?
Absolutely, yes. So as you can see, hopefully in my presentation, I mentored a number of students. They've done hands-on research with me, but I have a lot of teaching experience from my graduate days. So I taught a lot as a graduate student. I even developed a class as a graduate student from scratch and taught it on my own. Currently, I'm serving adjunct positions. So I'm adjunct at the College of Florida, where I taught intro biology with my colleague Brad Overley. And then I also teach biodiversity of Earth to art students at the Ronald College of Art and Design. So that's personally rewarding. It's not a scientifically rigorous class, as you might imagine, but I get to share my passion for biodiversity and conservation with students that may not hear it otherwise. So it's a very empowering class, and I think they get a lot out of it as well. Other than that, I have a lot of other teaching experience too. I do a bunch of guest lecturing, but yeah, brief answer. Yes.
So you mentioned the
Yeah.
Erisea aborigine.
Aboriginal.
There we go. So you mentioned that it needs to go through a digestive tract. Aside from the gopher tortoise, what other animals typically eat the seeds?
That is a great question, and all of that is anecdotal, right? So the reason that we think the gopher tortoise is because they share a very similar habitat. So oftentimes, the gopher tortoises, I don't know if you know much about them, but they burrow, right? So they don't like water, even though they live very close to the coast. So they burrow and they create these houses. And oftentimes you find Erisea aborigine right outside of their houses, right? We've seen evidence of what looks like a gopher tortoise chunk from the base of the cactus. So again, a lot of this is anecdotal. It could be that birds might try to eat them, but the fruit are pretty large. So I mean, it could be that the fruit falls on the ground and the birds can, dissect it that way and eat it. But yeah, I think they're tortoise, and that's kind of just anecdotal evidence of that in the literature as well. But if I could do a study, I would.
Thank you.
Yes.
Sally, you have one slide where you showed the different communified populations and you had stressed them. And it seemed like the New York population had the most tolerance.
Correct.
As you went further south, Alabama had less tolerance.
Correct.
Do you think that might reflect the adaptation to the local climates of Alabama? For a few winters down here, I would imagine, as opposed
Exactly.
to state.
Yes, so that's a great question, and it's very intriguing. So desiccation tolerance actually has a very similar, and I'm not a biochemist, but from what I've read, it has a very similar biochemical pathway as cold tolerance, right? So as you might imagine, cold environments are very dry, right? So being able to tolerate colder conditions might also mean that you can tolerate this desiccation tolerance a little bit better. So I think those two kind of go hand in hand, and you're exactly right. Yeah, it's a great observation. Yes.
I was curious about your New York site.
Mm-hmm.
Was that in Cattaraugus County?
That might be. I was in Olean State Park. Yes.
So you were in the unglaciated part of Cattaraugus County?
Yes. Mm-hmm.
So the bacteria seems to be restricted to unglaciated Northeast America.
Yes, absolutely. Yeah. So it's interesting. The northern range boundary, if you compare the glacial boundary line, it really matches up really well.
It was right south of that little river corridor in Cattaraugus County.
Yes. Yeah. And the interesting thing too is that there's another fern species that only exists as a communiphyte as well, that's found in these rock shelters too. It has distributions that extend well beyond that of the Tenure aboriginiana. So it could be that, and I don't want to get too much into this, but it could be that the sporophyte of, this is called Crepinemes, it could be the sporophyte of that species was able to handle the glacial climate a lot better than that of Botania. So it had sporophytes that kind of held out longer, and then spores that could, expand and then allow for, that range expansion further north compared to that of Botania. Great question. Yes.
So you mentioned that people don't really eat ferns. Is that true?
So, yes, so that's right. There are people and there are communities that eat fiddleheads. I personally have fiddleheads myself. It's very common in Asian culture actually to eat fiddleheads, but they also have high rates of stomach cancer. So I don't know, correlation causation, I don't know, but traditionally, compared to other angiosperms, other things like that, consumed fairly less. And even, delivery is not a huge concern for fern research as well. So I make my life a little bit easier in terms of ecology work.
Tell a general question, is it known why some fern gametophytes cannot produce a sporophyte?
That is not known, and that is something that everybody wants to know. And so before I came along, so Don Farrar is the scientist that did all of the work with these amazing rock shelter gametophytes, and he found one population in Ohio that had a very juvenile sporophyte, and it aborted. And there was someone at the University of Tennessee, and his name is escaping me right now, unfortunately. And he did manipulative studies where he basically threw a bunch of chemicals at bacteria to try to get them to induce sporophyte production. Same thing, they would produce juvenile sporophytes and they would abort. So we suspect there could be some kind of genetic thing going on. And while I love ferns, their genomes are insane. So trying to, get to that question, we need a lot more advancement in the genomic resources that we have. But that is the $1 million question. Everybody wants to know that one. Yes.
Why isn't herbivory an issue?
Probably because of a lot of secondary compounds and metabolites. Yeah.
Phenolics.
Oh, sure, yeah.
Yeah. Interestingly, I kind of have a side interest in like medicinal plants, and I never hear anything about ferns being used medicinally. And I was in Peru a few years ago doing some work down there, and I was working with some local organizations to help them identify ferns, so teaching them different characteristics and that type of stuff. And we come to this one tree fern, and one of the individuals was like, "Oh yeah, we use that for birth control." Right, so there are uses of it. It's just it's not widely available, and I think there may be one book published on it. Mark Plotkin, if you all are familiar with him, famous ethnobotanist, he gave a talk to Selby a few years ago, and I asked him, "Do you know anything about medicinal ferns?" He was like, "There's one book out of Colombia, and it's in Spanish," and he was like, "That's all I know." But he had this really great story. He was down working with a tribe, and he had a foot problem. And the shaman took a fern and threw it in the fire, and then took it out of the fire, put it on his foot, and then made a tea, and drank it, and his foot ailment went away. And he had been going to doctors, here in the States, and it wasn't, addressing the problem. And he said that the problem went away for a few months, and then it came back, and he went back to the same shaman, did the same thing, and he hasn't had problems since. So something's going on, Yeah, actually, that's
Any further questions for Dr. Chambers before she breaks for lunch?
great. Thank you all.